Welcome to our third podcast on lymphoid tissue, and within this unit we will cover the spleen and all of our different tonsils that we have in the body. So we're going to look, as we have with the other encapsulated organs of the lymphoid system, we will look at the structural components of the spleen, as well as the cellular constituents, in particular when we look at the red versus the white pulp in the spleen. And then we're going to be able to identify the different types of tonsils we have in the body. We'll have the palatine, the sublingual, and we will also have the adenoids, which are the pharyngeal tonsils. So the spleen is going to be the largest of all of our lymphoid organs that we have in the body, and it's located in the left upper quadrant of the abdominal cavity. I believe many of you have seen the spleen when you did your dissection on the cat. You can find it in the sort of inferior greater curvature of the stomach, where you're going to find the spleen on the lateral side, basically. When you look at the spleen, it's going to have a convex and concave surface, and the concave area is going to be the hilum. And this is where arteries and veins enter and exit. And this is not untypical of what we find with many of our organs, in which arteries and veins are going to leave. Certainly in the lymph nodules, we have that. The adrenal gland, we have that. The kidneys, we have that. So it's no different here. Since this is an encapsulated organ, we will have our dense irregular elastic connective tissue capsule, and to some degree we will have trabeculae diving in, as we've seen with our other organs, diving into, not necessarily to compartmentalize the spleen, but just to give it additional structural support. So here we see the spleen in the left upper quadrant as illustrated, and then we also see that we do have ligaments that are going to attach portions of the spleen back to our gastrointestinal tract. We will have a hepatic gastric ligament, and then in this case we are attaching this ligament that you can see in yellow to a portion of our colon. Now, the capsule will, in fact, extend downward into the deeper parts of the spleen as trabeculae, and these are predominantly going to pathways in which blood vessels are going to run. We will have trabecular arteries and trabecular veins, and the architecture of the spleen, by and large, are going to be maintained from reticular fibers, and these are our collagenous reticular fibers, and we're going to have reticular cells, for all intents and purposes, our fibroblasts. But in this case, since we have so many immune type cells, and we're also going to have a lot of red blood cells and platelets within this tissue, we're going to have to protect those collagen fibers, because that would start a platelet plug and a clotting cascade. So we're going to see how some of the cells of the spleen help to shield the reticular fibers that are there for structural support from the platelets themselves. Now, as we look at the spleen, we're going to see that we have venous sinuses. These are going to be some of the more important and characteristic parts of the spleen. We have our trabeculae, which we've mentioned, but we also have the parenchyma, or the cellular portion of the spleen, and this is going to be white pulp and red pulp. The white pulp are the leukocytes, and these are primarily our lymphocyte lineages that we're going to see, but we're also going to have macrophages here as well. And these are going to be surrounding many of our vessels that we're going to see. But then as we get into the area where we have these venous sinuses, this is where we're going to be passing red blood cells through these staph cells of the sinuses to clean them and to get rid of old red blood cells. And so the red blood cells will be spilling out into these sinuses, and that's where we'll find the red pulp. So in this illustration, we can see the outer connective tissue capsule, our dense irregular connective tissue, and you can see in this case somewhat discontinuous trabeculae. And again, this is more for structure than it is for isolation of certain compartments, as we saw in the lymph node and in the thymus. So we're all about blood vessels, either bringing in red blood cells or white blood cells into the spleen. And so our delivery of blood as it's coming in is from the splenic artery. Once the splenic artery enters the spleen itself, we are going to have branches that are going to spread out, and these are going to course into the spleen through the trabeculae, so we refer to them as trabecular arteries. Now, once these trabecular arteries leave the connective tissue of the trabeculae itself, and they will be reducing themselves in diameter as more and more of these branch off, then we're going to call these a central artery. And as these central arteries make their way in and surrounding the parenchyma of the spleen itself, they're going to be covered by lymphocytes. And so this is the periarterial lymphatic sheath, or PALS, and this is really going to make up our white matter that we see within the spleen. Now, it's a little bit of an oxymoron, because this white matter is going to be the darkest, densest color that we see in the spleen. With our H&E stain, it's going to be a deep purple to blue, and the red pulp is going to be a lighter color, more reddish in nature, but with white as well, and that's how we will distinguish the white from the red pulp. So from our central artery, which when it's covered with white blood cells, it's going to be the white pulp or the periarterial lymphatic sheath that's surrounding it. When we send branches off the central artery, those are going to be the penicillar arteries. And these penicillar arteries sort of is a global category, but they're going to have three different sections to them. First, we're going to see the pulp arteriole. This is going to be that first part as it branches off of the central arteriole, heading out of the PALS and into the red pulp. Then as we leave that and as we're getting higher into the red pulp area, we're going to be encountering somewhat of an enlarged region of this arteriole that's called a sheathed arteriole. And it's sheathed because it appears to be dilated because it's covered with macrophages. So we refer to this as a sheathed arteriole, a macrophage sheath, or a Schweiger-Seidel sheath, as it's called because of the people that identified this. Now, the last portion of the arterial system before we get to our venous sinuses are called terminal arterial capillaries, and this is the end of our penicillar artery category before we begin to dump our blood into a sinus so that the blood can be squeezed through and out into the pulp itself. So here we see our illustration. We have our arteries and veins entering at the hilum, and we have a trabeculae that's coming in. We lose the trabeculae, but here we have the central artery, and you see how they're illustrating it covered by all of these tightly compacted cells. This is our white pulp that we see in this region. Now, this is going to be a secondary lymph nodule. You can see the germinal center, and you can see the mantle or the corona around that. But following the central artery is our periarterial lymphatic sheath, and we see a little bit lesser density of cells in this marginal zone before we get out into the red pulp itself. Here you can see the distinction between the white pulp here and the red pulp here. So as we branch off the central artery, here's our penicillar arteries, and you can see that we've got this broad category. And so first, off of this penicillar, we've got our pulp arteriole. There's our sheathed arteriole, our terminal capillary, before we get into these really expanded, closed-ended venous sinuses. Now, you can see we really haven't done a whole lot of gas exchange, and so to call this venous, we're just calling this the terminal end before we enter into a venous, the venous circulation. So here's a different way to look at this. Here we have our trabecular arteries and veins, and we can still see this coming up into the trabeculae. Then as soon as we leave the trabeculae, we've got our central artery, and then we're going to have off the central artery, branching out of the white, we're going to have our penicillars coming up, and we don't have as much detail as we had in the others. But again, here you see the central artery. This would be a central artery. Then we've got the penicillars. And some of these that are coming out of germinal center, for instance, like you see here, these are often called radials that are coming up. And you see how it almost looks like spokes of a wheel. This would be more like the penicillar that's coming up off of this central right here, coming out of the white pulp, entering into the red pulp, and entering into our sinusoid system. Now, histologically, looking at H&E stain, we've got a lot of dark spots with lighter regions between. It's this dark material that is going to be our white pulp, and here this lighter reddish material is our red pulp. Now, within each one of these, and many of these look like a secondary lymph nodule, we're going to be able to see the corona or the mantle region, the germinal center. Each one of these nodules is going to have associated with it, here you can see a central artery. So here you see another, there's a central artery, here's one, there's a central artery. This one, it's not as clear. There may be something out of the plane of section here, but most of these will have that. And then here you see this connective tissue surrounding this larger diameter vessel. This is a trabecular vessel running through one of our splenic trabeculae. Again, here we have our white pulp, our periarterial lymphatic sheath, and this is in fact probably a primary lymph nodule, but here you can see our central arterial adjacent to our white pulp. Here we have trabecular connective tissue and a trabecular vessel inside of that. So white pulp, again, it's our periarterial lymphatic sheath, is going to be composed primarily of the T lymphocytes. But then when we get into those areas where we have our primary and secondary lymph nodules, we're going to see those as our B lymphocytes. Now, in the less dense marginal zone on either side of that, we're going to see plasma cells, T's and B's, macrophages, dendritic cells, and remember, macrophages and dendritic cells are going to be our antigen-presenting cells that are presenting material to our lymphocytes. So as we mentioned, we have the central arteries giving off the penicillars going to those sinuses, but in some of those areas, especially when we have those lymph nodules, that's where we see the radial arteries, and they're going to enter into the red pulp. And in this marginal zones where we're going to have some of these venous sinuses, but they refer to them as marginal sinuses. Their functions are the same as whether they are in the venous sinuses or these marginal sinuses. It's just a locational difference. Now, these sinuses, this is where we're going to see material being able to escape, cells entering and leaving. So it's in these regions, especially in the marginal zone, where we see antigen-presenting cells presenting their antigens, picking them up, presenting them. Macrophages are going to be screening any microorganisms, lymphocytes entering and leaving the bloodstream, and then lymphocytes, once we do pick up these antigen-presenting cells and they possess antigens, the dendritic cells are going to give the lymphocytes an opportunity to recognize that material and mount an immune response. So in this illustration, we have somewhat of a secondary lymph nodule. We have the lighter germinal center, our darker mantle region, and here we see these radial arteries extending up, and then out here where you see all the red, these are red blood cells, and so this is going to be our red pulp. Now, the red pulp, when we have these venous sinuses or splenic sinuses, these are going to be structures that are composed of cells, but we say they're barrel-shaped because we have these elongated endothelial cells called staph cells, and they're going to be lined up parallel to each other, and they're going to be wrapped with a couple of these perpendicular reticular fibers, much like the rings of a barrel. And because of the positioning of these endothelial cells, these staph cells, and the reticular fibers, there are going to be gaps between these endothelial cells, maybe 2 to 3 microns in diameter. And remember, red blood cells are about 8 microns, so it's going to be a very skinny, small space with which red blood cells have to pass through. Here we have a light level microscopic image on the left, and its corresponding electron micrographic image. And here we can see some of the splenic sinuses. That's what the S is going to be here. We have an endothelial cell that we see located here. Those are both of the E's and that S. So these are the corresponding spaces that we see. And so here we have the cells, the endothelial cells making up the sinuses, and here we see red blood cells in the center of these sinuses. And in some of these images, I'm not sure if we'll have them on here in lab, you can actually see some red blood cells being squeezed through these sinuses and out into the red pulp. You can see some red blood cells actually out here in the tissue. That's a red blood cell, and here's a red blood cell actually out in the red pulp itself. Here's our illustration showing the staph cells running in this direction parallel to one another, and then the reticular fibers wrapping them and holding them in place, and then here we have erythrocytes squeezing past those staph cells, being cleaned and screened, and if they're too old and too rigid, then the hydrostatic pressure pushing them through will actually shred them as they pass through these small spaces. Now, outside of the splenic or venous sinuses, we're going to have the cords of Billroth. Again, there are a lot of reticular fibers and reticular cells that are present to maintain the structure, but this is where we have a ton of cells. A lot of red blood cells, macrophages, plasma cells, lymphocytes, you name it, they're there. Now, the macrophages are there to all the cells that are destroyed, all the debris that's removed from the surface of the red blood cells, they're there to gobble it up. But the reticular cells are there to actually cover over reticular fibers and prevent starting a platelet plug and therefore the clotting cascade. So here we see in this illustration the red arrows are showing the direction of our splenic sinuses, and the green arrows are simply showing the space between those two sinuses. That's a cord of Billroth, and that's where we have all the cells, and you can see the greater density of cells outside versus inside the sinuses. Now, as red blood cells are squeezed through these small 2 to 3 micron openings, material that's collected on the surface of the erythrocytes it's going to be squeezed out or removed. Some equate this to squeezing a zit. That's kind of a nasty analogy, but it's kind of what it is. It's called pitting. However, old red blood cells are so rigid that they are going to absolutely be shredded as they pass through. But then again, as they age, there is going to be a loss of sialic acid on the surface, exposing galactose residues, and this is going to increase the phagocytic attraction of macrophages to these old, dead, and dying red blood cells. Now, as they're shredded, hemoglobin that's going to be taken up by the macrophages is going to be broken down into heme group and amino acids, and the heme group is going to be converted into bilirubin, which is conjugated to albumin, shipped to the liver, where the liver is going to process it. It's going to be conjugated to glucuronic acid, placed into the bile. Some is going to be removed in the gastrointestinal tract through the bile as stercobilinogen, or it's going to be excreted in the urine as urobilinogen. Stercobilin is the name of the fecal material. Urobilinogen gives our urine its yellow color as this is removed from the body. Now, too much bilirubin builds up in our system. That's when our skin, our eyes are going to turn yellow, and that gives us the condition of jaundice. Now, the last thing we have to deal with from the breakdown of red blood cells is transferrin. This is a protein, a serum protein, that is going to be used to coat the iron that's released from the heme groups as red blood cells are broken down, because iron is certainly not water soluble. So transferrin coats the iron, making that whole package then water soluble for its transport back to the bone marrow to be recycled and used to make new hemoglobin and placed into new red blood cells. Here we see a fairly familiar figure. We've seen this in physiology, but this is the breakdown of our red blood cell, and this is happening in the spleen. But these breakdown products enter into a macrophage. Here's our heme group broken down into bilirubin. Here we see our proteins broken down into amino acids. Iron bound to transferrin, it's transported. Bilirubin is conjugated to albumin that then goes to the liver and our hepatocytes, conjugated, and then through the various steps eliminated in the bile or through our urinary system. So that's the spleen until we at least look at it in lab, but we're going to finish off our encapsulated, or at least partially encapsulated, lymphoid tissue by looking at our different tonsils. Now, our tonsils are really going to be described based on their position. However, we're also going to describe them based on their histological appearance. And we're going to start with the ones we're most familiar with, and these are going to be our palatine tonsils. Now, these are the ones that if you look into your oral cavity, they're going to be situated between the palatoglossal and the palatopharyngeal folds. Essentially, on either side of the uvula, where you're going to see these enlarged structures almost look like the surface of a walnut. Now, as we look at those, we're going to see that their surface is stratified squamous non-keratinized epithelium. And if that sounds familiar to you, it should, because that's the same epithelium that we have lining our oral cavity. Now, this epithelium is going to dive down into, through invaginations, down into the tonsillar tissue. So we're going to be increasing the surface area with which antigens can be exposed to and contacted to the underlying lymphocytic tissue. And these invaginations are going to be called tonsillar crypts, and you may have upward of a dozen of these in a palatine tonsil. Now, the deepest part of our palatine tonsil is not going to be stratified squamous non-keratinized epithelium. It's simply going to be our lymphocytic tissue adjacent to a dense fibrous capsule. And so early on, removal of tonsils was often done with a wire lasso that you loop around the tonsil, and as you pull that tighter and tighter, it just simply slices through the dense fibrous capsule to remove that tonsillar tissue from the underlying connective tissue. Now, certainly as we look at the cellular component of the palatine tonsil, and all of these tonsils really, we're going to see a number of lymphoid nodules, primary but mainly secondary lymphoid nodules. So in this illustration, we can see quite a few of our lymphoid nodules, and these are our secondaries with their germinal center. Here we see the surface, and we see an invagination diving deeply down. A lot of time these palatine tonsils, these are going to be fairly large, and you can sort of guess this from the planus section, but it's going to have debris down in these crypts as well. This is why sometimes these get infected, you get inflammation, and you get tonsillitis. Now, if we could look even more closely at this, we would see that our non-keratinized stratified oral type epithelium is continuous with the surface, and then we have our dense connective tissue, dense irregular on this side. This is the free surface facing the lumen. This is the side contacting the body. Now, our pharyngeal tonsils are the second ones, and these are usually removed at the same time where our palatines are removed, and these are often referred to as the adenoids. Now, these are not in your oral cavity, but rather they're in the roof of the nasal pharynx. There's a thin capsule of dense irregular connective tissue, and instead of having our tonsillar crypts, these are going to have shallow folds they're referred to as pleats. So that's one way that you can distinguish histologically palatine from pharyngeal. But also, if we can look more closely, we're going to see that the surface epithelium on the free border is going to be pseudostratified ciliated columnar epithelium, and that is going to be the same as what we see with our nasal mucosa. So here's a location of our tonsils. These are our palatine tonsils. And then this is the location of our pharyngeal tonsils. Looking at this tissue, we can see our secondary lymph nodules. We can certainly see what appears to be a stratified layer. However, as we look closer and closer at this, we're going to recognize this is our pseudostratified ciliated type of epithelium. We see these invaginations, or in this case, the pleats are folds. We're at much higher magnification than we were earlier looking at our tonsillar crypt. Once again, secondary lymph nodule, lymphocytes. Here, I think we can begin to get a feeling they were pseudostratified, and then we've got our fold or pleat present in that tissue. Now, if we're able to look at high enough magnification, certainly here we can see that we definitely have pseudostratified layer, and here we've got a nice ciliated layer with this nice dark line. Do you remember what the dark line represents? Think back to our epithelium. Basal bodies. These are the basal bodies of the cilia lining up. So it's important to look at all those things and really understanding which tonsil we're looking at, because they're all going to have the lymph nodules. Now, the last are the lingual tonsils, and these are going to be on the lateral surface of the tongue. They say the dorsal surface, and so, yeah, but really we're talking about the side of the tongue where you're going to see these. Stratified squamous non-keratinized epithelium, certainly our oral type epithelium underneath. We're not going to have much in the way of the capsule, and all we're going to see with our lingual tonsils is a single crypt. So in this case, looking at somewhat a plane of section, looking at the magnification, this is really the only way we're going to be able to tell if we're looking at lingual versus palatine tonsil. So here we have the dorsum of the tongue where we have all of our papillae. Here we have the ventral surface, and right on this sort of more distal posterior third of the tongue in the back, and right on the edge, we're going to see these folds that are going to be our lingual tonsils. Now, in this case, this is a little bit tricky because this is going to be our tonsillar crypt. All of these things that you see, these are artifacts. You can see how they look jagged and cracked in this particular one, but this is one of the best lingual tonsil sections I could find. Again, with the plane of section, with the magnification, you can see our secondary lymphoid nodules. You can see our tonsillar crypt. We're not seeing a lot of anything else that's really going to give us a clue. So I'm going to have to clue you in as far as this is the tonsillar crypt or one of multiple tonsillar crypts for you to know whether this is going to be lingual or whether this is going to be palatine tonsil. Again, here we see the surface, and once again, we're going to see our non-keratinized stratified squamous, exactly the kind of epithelium we're going to see on our palatine tonsil.